Okay, everyone, welcome back to part 2 of the Metabolism and Nutrition chapter. In part 1, we had the lovely joy of giving some basic definitions. We talked about the definitions of metabolism, both anabolic and catabolic reactions. We talked about cellular respiration, enzymes, coenzymes. We talked about the 3 energy systems that cells utilize, the phosphagen. We talked about the anaerobic metabolism and aerobic metabolism. And then lastly, we began our discussion of the specific process of cellular respiration, converting glucose and fat and protein, sometimes, into ATP, because remember, ATP is the usable energy source. You may recall our discussion about glycolysis. As a matter of fact, just because I like you all so much, let me go back and just review what we talked about. So we said the first series of reactions in cellular respiration are called glycolysis. In this process, we convert glucose into pyruvate. For every 1 glucose that comes in, we produce 2 pyruvic acids and 2 ATP. Some of the other characteristics of this first phase, it occurs in the cytosol. It does not require oxygen. It involves chemical reactions known as phosphorylation. And one of the important enzymes that's involved in this phase is called pyruvate kinase. Cool. Now, if oxygen is present, remember, if oxygen is present after glycolysis, we swing right on over into the Krebs cycle. But if oxygen is not present, there are other options. You don't have to know the specifics. But one of the downsides of when we stay in glycolysis too long is that we can build up excessive levels of lactate, which can make the blood acidic. Okay, by and large, most of the energy that our cells produce, meaning most of the ATP that our cells produce, occurs through aerobic chemical reactions, which are going to include the Krebs cycle and the electron transport chain. Glycolysis always has to happen first. We need to have a means of getting quick energy by using glucose and producing 2 ATP, but that is not going to be very efficient. Once we get the benefit of oxygen, we can utilize these last 2 aerobic means and we can produce many, many, many more ATP. All right, so we started by talking about glycolysis. Glycolysis left us with 2 molecules of pyruvic acid. So that pyruvic acid that we formed from glycolysis enters the Krebs cycle. That pyruvic acid will then be converted into acetyl coenzyme A, which then gets converted into the end product of the Krebs cycle, NADH and FADH2. For our future exams, there's not going to be an essay, but you can imagine where I have you put the steps in order. So for example, for the goal of the Krebs cycle, I would like you to know that it starts off as pyruvic acid, then it goes to acetyl coenzyme A, and then it goes to NADH and FADH. You can even have a way to remember that. P for pyruvic acid, A for acetyl coenzyme A, and then N for NADH and FADH, PAN. Other things to know about the Krebs cycle. Now we're in the mitochondria. Remember, glycolysis happened in the cytosol of the cell, but now the Krebs cycle chemical reactions are occurring inside the mitochondria. It is aerobic, and it has 2 phases. The first phase, if we look at the top, we can really figure this out. Let me go back to the red. The first phase is converting pyruvic acid into acetyl coenzyme A. So the first part of the Krebs cycle is doing the first thing, converting pyruvic acid into acetyl coenzyme A. And then the second part, which is going to involve the Krebs cycle, is converting acetyl coenzyme A into these mitochondrial enzymes. And the second part gets its name because it occurs in a cyclic reaction. I think it's funny when I use this program and then I click done to go to a next slide. Sometimes part of the pen markings bleed. So look at the A in pen. It kind of looks like a 90s rock band or a 90s grunge band label with the bleeding A. I digress. Here is actually a pretty nice visual. Sometimes I feel like visuals get too complicated and it can be hard to lose sight of the whole thing. But why I like this visual is because it shows us the two parts of the whole of the Krebs cycle. As we said, the first part is we have pyruvate from glycolysis. And the first part is that we convert pyruvate into acetyl coenzyme A. So that's the first part. The second part is that we take that acetyl coenzyme A and it goes into a cycle. Hence we get the name Krebs cycle. And what are the end products of the Krebs cycle? What's being kicked off of these cyclic chemical reactions? FADH and NADH. And this follows along with what we talked about here. The first phase was converting pyruvic acid into acetyl coenzyme A. Right? The first phase was converting pyruvate into acetyl coenzyme A. The second phase was converting acetyl coenzyme A into NADH and FADH. The second phase is part of the cycle, converting acetyl coenzyme A into what's being kicked off of these chemical reactions or released, NADH and FADH. How about that? You don't have to know the specific numbers. I'm not going to ask you whether it's 8 or 4. You don't need to know the 2 ATP either. I mean, it's true, but I'd rather stick to the other facts. All right. Here's a simplified view of what I just said, right? Phase 1, pyruvate into acetyl coenzyme A. Phase 2, that acetyl coenzyme A enters a cycle where we produce NADH and FADH. As I did before, I've included my notes. You don't have to know these specifics, but it goes through the same steps. What's step number 1? Converting pyruvate into acetyl coenzyme A. Boom. What's step 2? A cycle of chemical reactions. And this is where it gets crazy because there's 10 steps. So now we can see the one side of the Krebs cycle. Steps 1, 2, 3, 4. And then we can see the remaining step, or 9 steps, 5, 6, 7, 8, 9. Whew. And here it is all together. Sometimes you think back and you're like, how did I memorize all that stuff? Slowly. Okay, what are some other key points of the Krebs cycle? Well, lipids, this is where lipids will come in. So we've been talking about glucose. Glucose comes in. If we think about glycolysis, we started up here with glucose. Glucose came in and it formed pyruvate. And then pyruvate is formed into acetyl-CoA. And then acetyl-CoA enters the Krebs cycle. Well, this is where lipids come in. So just like we can metabolize glucose for energy, we can also metabolize part of the triglyceride lipid for energy. And I would like you to know that where do lipids come into this process? They enter the Krebs cycle. So just imagine if a lipid's going to come to the party, that lipid's going to come in at this step right here. And then the rest is the exact same as it would be for glucose. Something else I want to point out, about the Krebs cycle is that we can see here where so much of that CO2 is produced as a byproduct. If I go back up to my crappy pencil drawings, we're going to notice that you see these byproducts in blue, and we can see here we kick off CO2 here. We kick off CO2 here. So I'm showing you, we've been saying that CO2 is a waste product. And now we're seeing where. CO2 is a waste product of cellular respiration. So every time we're metabolizing glucose or fat for energy, every single time, we're releasing CO2 as a waste product. So just like we need to bring in oxygen to allow the Krebs cycle to happen, because it's aerobic, we're also releasing CO2, that's shown over here. We're also releasing CO2 as a waste product, which is something we've got to get rid of from the cell, put back into the bloodstream, transport it back up to the lungs or to the heart, send it to the lungs, and then take it back up the airway so we can exhale it. I've given you one key enzyme for the Krebs cycle, but I'm not going to ask you to know it. It's not in green. And the second point, you can see it's on here, so you can read it, but I'm not going to ask you this on the test. If you're interested, even though we say that the Krebs cycle is aerobic because it needs oxygen, and that's true, but it's actually that oxygen is used indirectly. Oxygen is used indirectly to oxidize NADH back into NAD+, which is one of the parts of the Krebs cycle. So it's more of an indirect use, but for us, it's not a point you need to worry about. All right, so if I want to review, because I think it might be good, just like we did for glycolysis, if I review the most important parts for the Krebs cycle, we're going to say that it needs oxygen, it occurs in the mitochondria, it is the second phase, so it happens after glycolysis. What happens in the Krebs cycle? We have pyruvate from glycolysis, and it is converted to something called acetyl coenzyme A. And this is actually the first part of the Krebs cycle, converting pyruvate into acetyl coenzyme A. And then the second part of the Krebs cycle is that acetyl coenzyme A enters that cyclic series of chemical reactions, and it forms NAD and FAD. Actually, let's just go back for 1 second, because I want to make sure that we're specific. We've been talking about both. Let me go all the way back. Excuse me. Part of this process, you're going to see NADH, FADH, but there's also NAD and FAD. What I want to point out is that the end products of the Krebs cycle have the H on them. So let's go back to my review. Excuse me. So we had pyruvate being converted to acetyl-CoA, and then acetyl-CoA converted to NADH and FADH. And that's the second part of the Krebs cycle. And this is the more cyclic part, because it's a series of chemical reactions. Other things that we talked about. The Krebs cycle is where lipids enter. If I can spell. I can't have that be on record. This is where lipids enter. Something else we talked about is CO2 is produced as waste product. Okay, so even though these are complicated chemical reactions, I'm trying to be very clear about what you need to know and what you don't need to know. All right, now we've finally gotten to the last phase of cellular respiration. We started with glycolysis, which in the presence of oxygen led us to the Krebs cycle, and the Krebs cycle leads us into the electron transport chain. What are some things to know about the electron transport chain? It is the last step. And what are we going to form? ATP. The whole idea of this was to start with glucose in glycolysis. After a bunch of chemical reactions, glycolysis, Krebs cycle, electron transport chain, the goal is to form ATP. Where is this happening? Well, not only are we in the mitochondria, but we're in the mitochondrial cristae, which is a fancy word for folds. You may remember the mitochondria. It's kind of oblong, looks like a sausage, and it's got these folds on it. Okay? Those folds are the cristae. So when we did, I'll use red here, the Krebs cycle is happening inside the mitochondria, but not along the cristae. But it is the electron transport chain that is happening along the cristae. Okay, so what I'm drawing here, this is where the electron transport chain is happening. We have to be more specific. It's not just happening inside the mitochondria, it's happening along the mitochondrial cristae. Other thing I'd like to point out is similarly to glycolysis, we have some phosphorylation, but more specifically, the electron transport chain is involving oxidative phosphorylation. All right, good stuff. Let's talk about how it happens. We can see here some simplified views. It's kind of simplified, but it's still pretty complex. I've also included my own notes. So for those of you, like I said in the first video, I found it difficult with the textbooks and with all these things to really break it down. So for me, it was very helpful if you ever needed to learn this more specifically, to make my own notes and break it down into steps that I understood. What can we see from this picture though? We can see that the steps of the electron transport chain are happening along the cristae or the folds, right? So we basically have a series of chemical reactions that are going into and out of these folds. All right? You do not need to know these specifics. And here's part 2, because the fun never ends. What is the overall goal of the electron transport chain? To form ATP. I even highlighted it because I was so excited. Okay, so I've tried to break this down in a simplistic way that allows us to understand maybe a little bit of what's going on without having to get too far in the weeds. I've basically put it into 2 phases, and I think that this might be a reasonable way of understanding a little bit of it. So let's talk about the electron transport chain. Maybe we could say that the first phase of the electron transport chain is taking the coenzymes that we got from the Krebs cycle. What was from the Krebs cycle? We had NADH and FADH. It's actually FADH2, but we don't need to be that specific. Everything is successive. It's just like a row of dominoes. All the steps have to happen beforehand to get us here. So if the Krebs cycle happens as it should, the end products of the Krebs cycle are these coenzymes, NADH and FADH. The first phase of the electron transport chain is that these coenzymes release electrons. Now, wait a minute. You're probably saying there by yourself, but Dr. Jenkins, I remember. What is the job of a coenzyme? There's a method to my madness, ladies and gentlemen. We talked about coenzymes. What was one of their function? What was their function? To transport or carry electrons. So up until this point, these coenzymes have been carrying electrons. And part of the name of the electron transport chain is because these coenzymes carry or transport electrons. Eventually, we want these coenzymes to release the electrons. And after that happens, through some other wonderful chemical reactions, we produce oxygen. So we're going to break it down. That's going to be our first phase of the electron transport chain. And let me go back to the simplistic view, because look here on the left. Let me do it in, I think red will be good. So we have NADH and FADH2 from the Krebs cycle. And what are these little guys doing? They're carrying electrons. All right, so these come in from the Krebs cycle, and they're carrying their electrons down this chain. That's why this picture looks like that. They're carrying electrons down that chain. But eventually, they release their electrons. And because those electrons have been released through the magic of more chemical reactions, we produce oxygen. So that's how we're going to simplify it. NADH and FADH from the Krebs cycle enter the electron transport chain. They carry electrons for a while, and then they release the electrons. And the release of the electrons later leads to the formation of oxygen. Right? NADH and FADH carry the electrons for a while, but then they release the electrons, which eventually allows us to form oxygen. Okay, now we get to phase 2. For phase 2, let me go back to black. For phase 2, we're going to be utilizing a concentration gradient. And I bring this out because, God, I've been saying this from chapter 1 of A&P 1. If you've had me before, you're probably tired of it. But it's almost like in every nook and cranny of our body, we're utilizing such simple things as diffusion. And that's what's happening here. We form a concentration gradient, and we have a semi-permeable membrane, and voila, we can utilize diffusion. So what's going to happen in phase 2 is we have a buildup of stuff on one side. It's technically protons, but I don't want to get that specific. We have a buildup of stuff on one side, and that forms a gradient. And by the way, one side of what? The mitochondrial cristae. So now we're operating along the cristae. That's why it's beneficial to have the cristae here. Let me go to red now. We have a bunch of things accumulate on one side of the cristae. And actually, get this to work here. Oh, can I get this to work? Doesn't want to work here. That's okay though. Let's go back to the red. We can understand that this membrane is semi-permeable, right? So it's actually like this. It's a phospholipid bilayer. All right. So we have a buildup of stuff on one side of the cristae. And when we have a gradient and a semi-permeable membrane, what's going to happen? We're going to diffuse from high concentration to low concentration. And guess what? That push, that energy that we produce simply by having a gradient is the energy that's needed to convert ADP, which is adenosine diphosphate, and we add that final phosphate. We had adenosine diphosphate. Now we add a third phosphate and we get ATP. Wow. Diffusion. Who would have thought? I'll tell you what, I thought. So if I'm using this as my study tool, and we say during phase 1, right? This over here is phase 1 of the electron transport chain. First of all, what are some things about the electron transport chain? It needs oxygen. It's the last or third phase. It occurs in the mitochondrial cristae. What is the overall goal? To produce ATP. Cool. How does it happen? Well, we have our first phase. We bring in the enzymes, the coenzymes from the Krebs cycle, which carry electrons for a while, and then they release the electrons, which eventually forms oxygen. Then we have our second phase. We have a buildup of things on one side of the membrane. So let's say we have a buildup of things on one side of the membrane. That develops a gradient. And we have a semi-permeable membrane. So the gradient, we move from high concentration to low concentration. That gradient is the last energy push to take what we had up to this point, adenosine diphosphate, and we add our third phosphate. And that is this movement right here. This movement is the energy to add that last phosphate, which yields ATP. That's how I would study if I were you. Okay. You can get more specific if you want, but you don't have to. I've given you the information about the key enzymes, but you do not have to know them. If you're curious in the electron transport chain, these key enzymes are basically inside the cell membrane, inside the mitochondrial cristae. So this is our enzyme complex number 1, literally inside the cristae. Here's our enzyme complex number 2, number 3, and it goes all the way up to number 5. But you do not need to know those. Okay. Whew. Now that we've talked about the meat and potatoes of cellular respiration, the rest of this chapter, we're going to be a little bit more general. Okay, couple points here. Is our body very efficient? It's more efficient than man-made machines. So Homer Simpson's man-made machine here may only capture 5 to 10% of the energy. So man-made machines are less efficient than us. So we are more efficient than man-made machines, even if we rounded this up to 40%. Our body processes are more efficient than a man-made machine, but we still lose a lot of energy as heat. Okay, so even though we're more efficient than man-made machines, it's just the nature of the beast. Anytime we have all these chemical processes involved in making something, there's going to be some heat and some energy lost along the way. I'd like you to have an idea about that number. We can recapture about 40% of the ATP. Okay, what happens next? Well, ATP has to be used, or that's it, we can't store it. So once we produce ATP, we quickly use it. If we have any excess, we got to break it back down into parts that we'll have to later put back together again. If we have excess nutrients, glucose and fat, we can simply store them for later. Okay, I'd like you to review these terms. And yes, they sound alike, but they're also important because we've already been talking about this. Now I'm just giving you the fancy names. We're constantly storing glucose as glycogen, and then we're constantly, when we haven't eaten for a while, for example, taking that stored glycogen and converting it back into glucose. Well, all these processes have names. So when we store glucose into glycogen, that is called glycogenesis. I think about this word, I break it down. Genesis means the production or beginning. Think of the first book of the Bible, to produce. And glyco means glycogen. So if I'm producing glycogen, or the beginning of glycogen, I'm storing glucose as glycogen. The alternate process where I break down glycogen back into glucose is glycogenolysis. And of course, these actions are triggered by hormones. Anytime that we do glycogenolysis, it's happening because of the action of glucagon. Anytime we're storing glucose as glycogen, it's because of signaling from insulin. Okay. Now, let's go back and talk about the other nutrients. We mentioned fat briefly, but we're going to talk a little bit more about the process. As we said, I was talking about glycolysis, the Krebs cycle, and the electron transport chain from a standpoint of glucose, starting with carbs and going all the way down. It's of course possible for fats and proteins to enter this process. Okay, I first want to point out a couple points, and I've already said this before. Fat can only be metabolized once we have oxygen. We cannot metabolize fat anaerobically. Once we do metabolize fat, it gives us the most energy per gram. 1 gram of carbohydrates yields 4 kilocalories of energy, but 1 gram of a triglyceride can yield 9 kilocalories. So once metabolized, fat can give us the most energy per gram. Know that number. But fat metabolism is not easy. We would like it to be easy because it gives us such good energy, but it's not. It's complicated. There's actually 2 ways that we can convert lipids into energy. You don't have to know the bottom one. Let me see what I wrote down here because I forget. I was being specific now, wasn't I? I sure was. We're going to focus most on the first one. Okay, just want to make sure I'm consistent with what I talked about. So we're going to focus on this one. Even though this is true, you do not need to know it for this discussion. So how do we talk about fats? In our previous discussion, we talked about how lipids can enter the Krebs cycle. And specifically, it is the fatty acid portion of the triglyceride that is able to enter the Krebs cycle. You may remember, we can't forget it, every triglyceride has 2 parts, a glycerol molecule and 3 fatty acids. It is our fatty acids that enter the Krebs cycle. This part you don't need to know. It is the fatty acids that enter the Krebs cycle. How do we do it? How do we get those fatty acids to enter the Krebs cycle? There's a bunch of steps. And actually, I wish this would have came out, but auto format. 1, 2, 3, 4. Fats are complicated. 5, 6, 7. Oh my goodness. I've given you my notes when I had to learn this. I mean, to put it bluntly, fat is difficult to deal with. It's not water soluble. It's a pain in the butt. You do not need to know all those specifics. I've picked out a couple things in green that I'd like you to know. What are some things that are unique about fat? One of the things that is unique about taking the fatty acid and trying to metabolize it for energy, we know that fats require some special care when they are circulating in the bloodstream. Because fats are not water soluble, they need a carrier. Glucose can just be transported by itself, but fat cannot. All right, so in order to be circulated in the blood, because lipids are not water soluble, they can't dissolve in the plasma, triglycerides need a carrier. And most often, they're going to be carried by that plasma protein called albumin. And if you think back to our first chapter this semester, the blood chapter, we talked about those blood proteins. One of them was albumin. One of its jobs is to be a transporter. So that's unique to fats. But where does the action really happen? You know, it's kind of like back in the day when they had that MTV show Cribs, and they got into the bedroom and they said, "This is where the magic happens." Like, you didn't think they were going to say it every time. Beta oxidation. This is where we convert the fatty acids finally into acetyl coenzyme A. It's through a process called beta oxidation. Boom, because once we form acetyl coenzyme A, that can enter the Krebs cycle. If you remember, we had two parts of the Krebs cycle. Converting pyruvic acid into acetyl coenzyme A was the first part. And then once we have acetyl coenzyme A, then it can enter the Krebs cycle. Oh, man. So beta oxidation, that's where we actually convert the fatty acids into acetyl coenzyme A. And it is important because it is acetyl coenzyme A that can enter the Krebs cycle. By the way, why is it called beta oxidation? Because it needs oxygen. Remember, we cannot metabolize lipids without oxygen. Just be reviewing what I talked about. Here's extra stuff if you want. A lot of steps. Oh, man. Okay. We have some terms for lipids too, breaking it down versus storing it. But I've chosen you have a lot to know, so you do not have to know these. They're not in green. I would like you to review ketones. If we thought about glucose, one of the downsides of burning excessive glucose through glycolysis was producing lactate. Remember that? And if we build up too much lactate, it can be acidic. Well, a similar thing can be true of fats. So when we metabolize fats for energy, and of course we're doing this in the Krebs cycle, one of our byproducts are ketones. And just like the lactate, ketones themselves are not bad. It's normal to produce ketones as a byproduct of fat metabolism. But ketones can be a problem when they build up in excess. Just like we can build up lactate and it can cause lactic acidosis, we can build up ketones and it can form ketoacidosis. One reason why a low-carb diet can be so dangerous, because when you don't have as many carbs and we're relying more on fat, you may think that's all good, but one of the downsides you got to be careful of is if I burn a lot of fat, I'm going to be producing a lot of ketones. Could turn the blood very acidic. This one you don't need to know about, remember? This one you do not need to know. Okay. We mentioned glucose, we mentioned fat. Let's talk briefly about protein. A couple of things to remember. This is the nutrient that we use last to metabolize for energy. Out of all of the energy that our cells produce, only 10 to 15% of the energy comes from protein. Meaning 85 to 90% of all of our energy comes from burning glucose and fat. We need protein to do its other jobs. Remember protein? Protein helps us give structure to your over 600 skeletal muscles. Protein helps to give us structure to our largest organ, the skin. Protein gives structure to hair, nails, ligaments, tendons. Protein we need to form a whole bunch of important chemicals in our body. All the enzymes, all of them, well, most of them, made up of proteins. Hormones, many of them, made up of proteins. Neurotransmitters, antibodies, and the list goes on and on. If we have to burn too much protein for energy, it means that we're taking away some of the benefits that protein can do with its other jobs. But we do burn some protein for energy. And to keep it pretty simple, a protein breakdown product enters the Krebs cycle. So very similar to fat. When we burn fat for energy, the fatty acid, it joins the party of cellular respiration in the Krebs cycle. And we can say, generally speaking, protein is the same way, enters the Krebs cycle. I do want to make a point because I have not talked about it yet. We did talk about glycogenesis, glycogenolysis. Make sure that you also review gluconeogenesis. And that's how I remember this, gluconeogenesis. Formation of glucose from non-carbohydrate sources. Even though I skipped this part of the lecture, I think it's an important term for you to know. Sometimes we can take the glycerol, part of triglyceride, and we can convert it into glucose. Or we can take a portion of the protein and form glucose. So gluconeogenesis, anytime that we form glucose from a non-carbohydrate source. And remember, I did want you to know glycogenesis and glycogenolysis. We did talk about glycolysis. I put this slide together because a lot of the words sound alike, but you didn't need to know the lipid ones. I'm trying to give you a break somewhere. Okay. We are almost done, but I would be remiss if I didn't talk about this. And I know that you probably don't want to hear it, probably thinking, aren't we done yet? But protein is a real pain in the ass, and let's just be honest. Protein contains nitrogen, and that is not true of carbohydrates and fat. If we think back to the basic molecular structure of carbs and fat, they contain carbon, hydrogen, oxygen. But protein contains carbon, hydrogen, oxygen, and nitrogen. And it is that nitrogen that makes protein a real pain in the ass. So this is saying what I just said, right? Carbs and fats do not contain nitrogen, but proteins do. Here's a nice little picture of an amino acid. And in that amino acid, we see carbon, we see oxygen, we see hydrogen, great. But we also see that nitrogen. And that's what makes protein so difficult for the body to deal with. How we get rid of nitrogen is very important. We have two mechanisms for removing nitrogen, because before we metabolize protein for energy, even though we do it in small amounts, we need to get rid of that nitrogen. Let me just peek ahead. Oh yeah. So it's almost like two steps. We've got to get rid of the nitrogen before metabolism. But then once we remove that nitrogen, how does the body get rid of it? So it's like a two-step process, this nitrogen. That's why it's such a pain. I think you're beginning to understand why. So before we can metabolize any part of protein for energy, we have to remove nitrogen. I'm only going to have you know one of these, deamination. There is another way, but just to make your life easier, deamination is one way that we can remove nitrogen before we can metabolize it for energy. And I chose this one because deamination, I think D helps me to remember like getting rid of something. If I detoxify, I'm getting rid of the toxins. If I deaminate, I'm getting rid of the amino group, which contains nitrogen. So just be able to know the definition. Deamination, process of removing nitrogen from the amino acid. Okay? Deamination, process of removing nitrogen from the amino acid. But now, even though we remove nitrogen from the amino acid, we still got nitrogen in the body. We just have it no longer attached to the amino acid. So we got to have a way to get rid of that nitrogen out of our body completely. and that is called the urea cycle. This is the body's most common way to get rid of nitrogen. And I mean to get out of the body. This occurs in the liver. So this picture, it may seem more complicated than it needs to be. But basically, in the liver, this is the liver, right? In the liver, ammonia is broken down into urea. So basically, we take the ammonia, which has the nitrogen. So when we deaminated, we removed nitrogen from the amino acid. We formed ammonia. Let me say that again. So when we deaminated, and you may recall, deamination is removing nitrogen from the amino acid. So now we've removed nitrogen, but it just so happens that our body, the way that it transports nitrogen is it forms ammonia. And it is only after we form ammonia that we're able to convert the ammonia into urea. Okay, so we convert ammonia into urea, which we take to the kidneys, and then we pee it out. When we get to the urinary system, we're going to see that urea is a very big waste product in our urine. Even though most of our urine is water, of whatever waste products are in it, a lot of it is urea. And by the way, we can deaminate not just proteins that needed to be used for metabolism, we can deaminate proteins that are muscle breakdown products. We can deaminate a bunch of proteins from all over the body. But because it contains that nitrogen, we pull the nitrogen off the amino acid and it forms ammonia. We convert ammonia into urea in the liver. And then it is the urea that we've broken down nitrogen. So now we no longer have nitrogen, but that breakdown product, urea, we send to the kidney and then we pee it out. And this is what's known as the urea cycle. We take leftover nitrogen, form ammonia, break that ammonia down into urea, and then we send urea to the kidneys so it can be peed out. I'll say it one more time. The urea cycle. Taking leftover nitrogen, forming ammonia, breaking down ammonia into urea. Then we send urea to the kidneys where it can be peed out. That's how we get rid of nitrogen. Okay, last topic. Whew. Okay, metabolic states and rates. So if we're speaking metabolically, and remember, metabolism is the sum of our chemical reactions. But if we're just thinking about kind of metabolically, what kind of state is our body in? We really only have two options. Sometimes I think people overthink this, and this is something that does not need to be overthinked. We have basically two metabolic states. We have an absorptive state, and we have a post-absorptive state. And guess what? We're usually either in one or the other. Okay? We have two metabolic states, absorptive and post-absorptive. Post-absorptive. We're usually either in one or the other. What is one? One is absorptive. Not surprisingly, this happens during and after a meal. If you think about a meal, you just brought food in, that's when you're likely to be absorbing the food. So the absorptive state is during and after a meal. Here's what I want to point out. When we are in an absorptive metabolic state, our rates of anabolism are greater than our rates of catabolism. In other words, our building chemical reactions are happening in greater amounts than our breaking down chemical reactions. This may seem counterintuitive because you may be saying, if I just ate, won't I be having a lot of catabolism because I'm breaking down nutrients? Well, sure, you're going to have some catabolism. But I'm just saying during the absorptive state, we have higher rates of building. And the reason why we have higher rates of building is because you now have the supplies. Because you have the supplies to build. You have just brought in some amino acids, some essential amino acids. You've just brought in glucose. Oh, here I am talking and I can't write at the same time. Because you have supplies for building. You just brought in glucose that is giving your cells a bunch of energy. So even though it's counterintuitive, just know that in the absorptive state, we're able to do more building, more anabolism. Of course, there's some catabolism, but there's more anabolism. And if you memorize one, you know the other by process of elimination. The other one is post-absorptive. This occurs when you haven't eaten recently, and the opposite is true. We're going to have greater amounts of breakdown. Why? If you haven't eaten for a while, you're going to have to do more breakdown of stored fuel. If I haven't eaten for a while, my body's going to be spending a lot of energy breaking down glycogen into glucose, breaking down adipose stores into fatty acids. So in a post-absorptive state, even though I'm doing some building, my rates of breakdown are greater because I'm spending a lot of energy breaking down stored nutrients for fuel. Okay, this is the last topic. This is actually the last slide. How about that? Thanks for staying along so well. Okay, total metabolism. We talked about that already, the sum of all the chemical reactions. If we were to make a pie chart, love me some pie. My favorite is cherry pie. If we were to make a pie chart of all the means of metabolism, make sure you know that our basal metabolism makes up the biggest chunk of the pie. And I would like you to know the percentages. Between, usually around 50 to 70% of all of our chemical reactions are happening as part of basal metabolism. What's basal metabolism? The energy needed for your body just to keep itself going. In other words, even if you laid on a couch all day, not that you would do that, but even if you laid on a couch all day, your body is still expending energy. Your heart's got to pump, your kidneys got to filter blood, your liver's got to filter blood, you got to do peristalsis. So basal metabolism basal metabolism are the chemical reactions just needed to keep your organs going at rest. I think it's surprising to many people that the largest part of your metabolism is occurring just to keep your organs going, even at rest. But if you consider all your cells working as tiny little factories, if you consider that your lungs and heart and kidneys and liver, everything has to keep going no matter if you're running or not. So basal metabolism takes up most of our metabolic activity. Physical activity is the most variable. To be honest, you don't have a whole lot of control over your basal metabolic rate. That's largely genetic, dependent upon age, gender. If someone is physically fit, it can improve their basal metabolism a little, but it's not going to vary a whole lot. What you have the most control over is physical activity. If you're more active, you're going to be burning more calories doing activity. So physical activity is the most varied, most variable. You have control over that. And usually it's between 20 and 35%. And the last thing is about 10%. The books will call it thermic effect of food. It really just means digestion. So no matter what, about 10% of all of our metabolic activity is being devoted towards digesting and breaking down nutrients. So know those percentages and be able to define each. All right, really well done. If you have any questions, I am here. Let me know.